Welcome to The Pitch. My name is Robin Young, and I'll be your moderator for tonight's program. As a reminder, this is a monthly program and is scheduled for the second Thursday of each month, 7 p.m. Eastern Time. At each program, two promising companies present their technology and business plan to our panel of experts. As for me, I'm the publisher of Orthopedics This Week, the largest publishing platform and most interesting publishing platform in orthopedics and spine. Let me begin this episode by asking a question that probably answers itself. Products that help the body regenerate tissue which has been damaged either by diseases like arthritis or by traumatic injury have a well-established market among spine and orthopedic surgeons, but they are often complicated to use and expensive. How popular could an easier, faster, potentially safer system be for creating three commonly used regenerative products? PRP, bone marrow aspirate, or plasma. ABC MedTech, which has a couple of longtime orthopedic and spine industry veterans running it, thinks their new product Optimus has those three attributes. Do they? Well, here is their pitch.
Hello, I'm Randy Allard, President and CTO, founder of ABC MedTech. I want to thank Orthopedics This Week for letting me be on the pitch. This is the Optimus Auto Centrifuge and Blood Concentrator by ABC MedTech. We're currently seeking $355,000 to complete our FDA study. The market is very large with increasing use in sports medicine and osteoarthritis injections. The market opportunity has been growing in ASCs. We see that key players have a large market share in this space, and also the market share is basically focused on ortho and neuro. As you may be familiar with, most competitors have complicated capital equipment and user interfaces. Our design is sterile, fully automated, and single use. We think we can provide the complete answer for a no capital solution. Our team is significantly capable in technology, biologics, and business. We initialized our first IP in 2019. And recent acquisitions show examples of some sales multiples. In summary, we think we have a strong IP position for a single-use sterile product in this field. And now we're going to show a short demonstration video. The user introduces 30 cc's of venous blood, or VMAC, into the central chamber of the Optimus unit. The separation process is automatically started by the user by turning the cover. It will automatically stop in 10 minutes with a light indicating the process is complete, and the unit will automatically stop. The unit can remain there for 4 hours with the separated constituents until the user is ready to use them. The user removes the cover, exposing extraction ports so that the user can pick the constituents that they desire. And in this demonstration video, we use artificial blood. That's why you don't see any separation. The constituent, such as PRP or plasma, is applied to bone granules or autologous bone graft for the user to use in surgery. Again, thank you for letting me provide this opportunity on the pitch, and we think that we provide a unique opportunity in this space.
Very interesting. I think we'll begin our questions today with Dr. Scott Bruder. And Scott, what questions would you have for ABC MedTech and their CEO, Randy Allard?
Hi, Randy. Excuse me. Thank you for sharing your story here and for walking us through. I have to say the video went by really quick. There are a lot of details that I would have otherwise be interested in asking. Let me start with Let me start with this. What can you tell us about the ultimate concentration of the platelets compared to your starting specimen? What kind of range concentration are you seeing?
So we completed a study in Australia with actual patients against a predicate, and we beat it by 42%.
Well, the predicate that may not be saying much because some of these predicates um, don't concentrate platelets.
That's right. Yeah,
So
well, we're within the range of a lot of the systems, commercial systems out on the market.
are you at least 4 times?
Oh, yes, yes, we are. Yes, we are.
Okay. And the maximum volume that you can use is 30 cc's?
That's correct. That's correct.
And how do you manage the differential between looking for platelet-rich plasma, or you said you can introduce bone marrow aspirate into the device when you're looking for a slightly different specimen?
Right. So the separation chambers separate the PRP into rings, and then the plasma's in the center. Bone marrow aspirate concentrate has similar hematocrit and should separate in the same manner.
But you're looking, in that case, you're most likely looking for the buffy coat.
That's correct. And you can directly see that. So with the lighted backlight and the visualization, the user or the surgeon can directly choose the buffy coat region that they want. They can directly see it.
All right. So he's or she is doing it by visual inspection.
Yes.
Right.
Yeah.
There's nothing automated about it.
No, it's not. They're looking through the top of it, and then they're choosing the buffy coat constituent at the ring that they desire.
Okay. So forgive me because these are sort of detailed questions, but
Sure.
but when you say that this is maybe the only, or you refer to this as a sterile disposable, there are other products out there on the market that are single-use disposable that don't necessarily have to remain in the sterile field for the entirety of the procedure. So are you saying that the yellow base and the disposable piece is sterile? The whole thing?
That whole thing, the yellow base, that's it. So the whole thing stays in the sterile field. With other systems, the cartridge or the syringe has to go outside the sterile field, and it gets exsanguinated through lumens or cartridges and then back through the syringe. But with our device, it never leaves that device. It's separated in the device and introduced in the device and taken out of the device. So there's no exchange to cartridges or syringes at that point.
So that yellow base that must have a motor and a
Yep, that's the whole thing.
That whole thing is disposable?
Yes, it is. Yep.
What's your COGS on that?
It's very reasonable. We are low COGS on this.
And do you think that your ASP is going to be similar, $250 to what these devices that are currently out there selling?
Well, when you say the $250, that's for like the aesthetics or the clinical settings, but there's also a different price point for ASCs and hospitals. And we've had it confirmed with some of the strategics that we are within that realm of what they charge now.
So BMAC is, the devices for BMAC are substantially more than.
That's correct. That's correct. Yeah, it's almost double. Yeah.
Even higher in some markets.
That's correct.
But in the setting where, are you thinking that you want to take this into market yourself and start selling this yourself? Because you're asking for $355,000 to get your 510K cleared. When you have your 510K cleared from a strategic standpoint, are you going to go to independent distributors and try and sell this? Or at that point, are you going to try to license or do some type of M&A work?
Yeah, well, we have a dual approach. We have the ability to do it ourselves and make some money, but we also have some strategics that are interested that when we get the 510K, we can go back and talk to them some more.
Yeah, I'm struck with the, so given that the majority, I'll finish this line of thinking, but given that the majority of use of PRP is in intra-articular injections for NEOA,
yes.
And what's the value proposition for having a fully disposable sterile unit that isn't used in a sterile field?
Right. Well, there's sterile techniques. So we have two approaches. We want to attack the bone reconstruction market for defects and trauma and things like that in a hospital, get the higher ASP. But then when we go into the clinic setting, we think we can show we have better quality because we're manipulating the blood and the platelets less. We're not pushing them through lumens or cartridges, and there's not as much lysing as we hope to prove. So we think that we handle the platelets more gently with our system. And so we can show a better quality in the clinic setting. That's what we hope to show.
Okay. And are you working with internal people to navigate your regulatory strategy, or you have
We have a regulatory person that's very knowledgeable in this area. And then the lab we're using in Australia has done several PRP systems, and they have a wealth of database knowledge for this.
Okay. Well, I wish you every success, and I'll pass it back to Robin to ask our other panelists to raise their questions. I could keep going for 20 more minutes because I've been working in this space a very long time.
Yeah, thank you.
Well, thank you, Scott. And that, again, that's why I started with you, because this is in your wheelhouse. So it's a great way to begin. So why don't I now go over to Mike? Mike, why don't you pick up the questioning, and then after you, we'll go to Audrey.
Sure. So thank you, Robin. Randy, can you kind of elucidate us or educate us more on the clinical experience you've had with this to date?
So right now we've done actual human IRB trials with human blood that's been donated. And then that blood's been concentrated in all the units. And then our testing lab has done p-selectin comparisons, viral aggregation comparisons, hypertonic stress. So we've looked at all the endpoints that the FDA is wishing to see. And so we're calling this a pilot study. But we also had a pre-sub with the FDA on what we need to do to finish the 510K submission. And really, they had no questions about our data points. They just wanted more units to increase the statistics.
So that sounded like you've taken human blood and spun it,
Yes.
but not re-injected anything. Is that
no? But the indication, the claim is to improve the handling characteristics of blood. So the actual 510K does not show the benefit of PRP. That would be in a white paper.
I'm not suggesting that you have to prove the benefit. I'm just asking, has any material from your device been re-injected or reused?
No, we don't have a 510. We don't have a 510K because we can't do that.
Okay.
Yeah. So that would be legal at this point.
In the U.S.
Oh yeah, in Australia. Yeah, right.
What about veterinary applications? Have you considered that? Is that an easier way?
There's a real benefit for canine ACLs and horse equine laminitis. It's a little bit lower price point, and so we hope to establish it in the human area and then kind of separate it off into the veterinarian space. We actually attended the ASCS in Las Vegas a couple of years ago to look at that market.
Interesting. And you're raising $355,000 to get through a 510K.
Yes.
Do you need money past? I mean, most companies would want money past that.
That's true. Yeah, that's true. Yeah, we have a pro forma that also shows an alpha launch, and it also shows all the money we spent to date. But that seems like the most important milestone to get us past some of the questions with the strategics is to get that 510K.
Yeah, my advice, to you might be that most investors would probably want, if they are interested in investing, would want you to have more, just so that you don't get.
We have that. Yeah, we have that. We have that available.
And have there been publications based on this yet? Any peer review publications?
No, we haven't. We want to get the 510K and then use our resource in Australia to do white paper research based on a university, driven research. So. You
want white papers, or do you want peer-reviewed publications?
Well, we'd start with white papers, and then going to the Biological Association meeting in Nashville. That peer review cycle is a little tough. So osteoarthritis, has shown a promise recently, but Scott can talk to that. Efficacy is a different level for PRP. Yeah.
I've been involved in some OA companies.
Yeah.
It was a painful experience. Well, look, Scott, Randy, good luck. And I think this is cool. I've always liked it. I like the idea of being self-contained, and I like the idea of keeping everything in the operating room versus, these concepts of taking things out of the operating room and growing it and bringing it back. So, congratulations, and I'll be interested to see how this goes for you.
Yeah, thank you.
Great, thank you, Mike. That was very interesting. And yes, OA can be painful for sure. Audrey, let me turn the program over to you.
Thank you, Robin and Randy. It's so nice to see you and hear about what you're working on. Kudos to you and your team for what looks like a simple and elegant solution that will improve quality. So I've got a couple. These will be kind of quick questions, but in a variety of areas. first off, what is the infection rate today in PRP? Is it known? Is there a range that you could quote?
You said infection?
Yeah.
No, there's just the risk of injection. Really, there's no infection rate. There's intrasurgical infection rate, which can be 30% in a hospital. So PRP doesn't cause infection. It's just a surgical site infection in hospitals. And so to bring a patient back to the hospital could be $60,000, to address that surgical site inspection. So it's somewhere around 30% that we think we can mitigate.
Okay, so I guess I was imagining that one of the benefits of this would be a reduction in infection since everything's happening in the sterile field, but it sounds like that really wasn't one of the driving problems that you were looking to solve.
It's part of the value proposition. We hope to reduce surgical site infections, but also show that we have better PRP that's more readily available with less intervention, because the nurse or anesthesiologist can use this and not have the rep or somebody else go out down the hall and concentrate this and push a complicated program. So we think ease of use is a value proposition too.
Yeah, for sure. So another kind of quick one. I didn't see a power cord. Not sure if I would have. There was a cloth there, but is it battery powered or electric powered? Battery powered. And I noticed that the device itself was kind of shaking. I know that might be normal when watching a centrifuge.
That's aliasing from the video. It doesn't shake. So that was just aliasing from the video capturing it. So it runs very smoothly. You can't even tell it's running.
Okay. So I'm curious what's left to do to prepare for the submission.
Yeah, so we did one submission. The FDA came back with more questions.
Uh-huh.
So we need to build more units based on their questions. We found some manufacturing improvements. We need to build some more units and go back and complete the IRB in Australia, and it's still open. So we'll enroll patients, build more units, and complete the 510K. So the pre-sub with the FDA has outlined a clear path to success. So we know what to do in the protocol.
That's great. That's great. And could you talk a little bit about the sales strategy, like your first year? What's the approach?
Yeah, so we can get a couple of distributors that want to put in their bag. It'd be an easy sell to carry along, but we see ASCs as a real key because they don't have to do any bioinstrumentation validation. They don't have to have any capital equipment. We've been in surgery over the last couple of years on another product, and there has been a line at the centrifuge in the hall. So you don't have to go find the centrifuge in the hospital or compete with it. So it can really be its own product on the shelf waiting to be used. And then once we establish those markets, we want to go into the clinical settings for the osteoarthritis injections, aesthetics, all the other benefits of PRP is shown. And that would be a lower price point, but hopefully more volume.
Okay, great. And are there reimbursement codes that will accommodate your larger price?
Yeah, well, the reimbursement code doesn't pay for the device itself. It pays for the doctor extracting, and manipulating. It's paying for the labor. So right now there's no reimbursement codes that pay entirely for it. So it's still a cash system in the injection market.
Okay. And do you foresee, this last question, do you foresee that this would be used in areas outside of the musculoskeletal field, like cosmetics and other? Yeah.
Definitely. We've been talking with military people to harden it and use it in those areas where electricity is not available, and also underdeveloped companies. Go with a GPO or something that would help get it out there where electricity is not readily available all the time, in field conditions. So yeah, we see those benefits in this.
And would it be the same design or modified?
Well, for military, we have to harden it. So we'd have to make sure that it can be dropped out of a helicopter and still work, but it'd be the same design. Right now, it's pretty robust with the cover and everything. It can be dropped and still work.
Great. Thank you, Randy, and congratulations again. Look forward to hearing more.
Audrey, thank you for that question. So I'm just sort of imagining the sales, drop from 10,000 feet and, takes a licking and keeps on ticking. Actually, I'd like to, if you don't mind, Scott, excuse me for picking on you again, but I have a hunch you may have a question or two follow-up. Please go ahead.
Yeah, I'm really fascinated by your strategy here, Randy, and with the changing landscape that the agency has now moved these blood products from CDRH to CBER.
Yes.
Did you get caught in the middle of that at all? Would you pre-sub with CDRH, or was your initial pre-sub and your initial submission also with CBER, so that you?
No, it was with CBER. We didn't go to CDRH. They pushed us to CBER. So that's why all the questions are based on the biologic side about the P-select and aggravation activation. Nothing really about the device itself. So we're in the CBER world of the biological, and so that's why they wanted more statistics on each one of those endpoints.
Right. Is there a reason? And I don't want to name anybody, but I probably know who you're working with in Australia. Does a lot of work for companies.
Yes.
Is there a reason? And a lot of respect for him. Is there a reason you went all the way there instead of doing it here in the U.S.?
Well, really, it was the IRB is a little bit easier over there, and also he's done a lot of the PRP systems going way back. So if we got into deliberation with the FDA saying, well, really, what about this data point? He could pull up his data and research on other PRP systems saying, really, this number is not real, or this is what it should be. So we think we have a real advocate in background data if we got into, discussions with FDA on numbers.
Yeah, I'm a believer in the use of PRP for certain indications, and that there are differences between high leukocyte and low leukocyte PRP,
Yes.
and that there are differences in concentration and absolute numbers. But as you know, you can't market for any indication beyond that it may improve handling of bone graft materials.
That's correct. And so we also have to do a handling study. The FDA asked us to do that. So we're actually squirting it on bone and doing a handling study.
That sounds like a human factor study. Is it a human?
They're asking us to do it.
That's crazy.
We're the first guinea pigs. So thank you to everyone else. So yeah.
Wow. I would have pushed back.
Yeah. It's been tough because we launched this during COVID. So we've had 2 years of really hard deliberations in getting this launched.
Yeah. Okay, lots of luck to you. Lots of luck.
Thank you.
Thank you very much, Scott. That was fascinating. And now we're going to turn it over to our audience. We're going to put up our poll. Is this a game changer? Is ABC MedTech a game changer? Will it challenge standard of care? Or is there more work to do? Audience, let's put up the poll. Good. So it looks as though about a fourth think it's a winner and a game changer. 3/4 think it could challenge the standard of care. Wonderful. Well, thank you to the audience for participating tonight. That was really interesting. I think both companies were outstanding. And with that, I will wish everyone a very good evening. Thank you.